Hey guys, I am so honored to be here amongst everybody doing these MAVE experiments. And I don't know if you guys know, but these right here, we have like the heroes of combining imaging and genomics, like Ann Carpenter and Doug and JT and Sriram. It's just such a pleasure to be here amongst you guys. And like you said, I hail from WashU in St. Louis, the MGI. And my name is Willie. And I kind of come from a neuroscience background and then an engineering background. And genomics is just such a good draw to people that have some of these computational skills, and I think just such a cool way to bring it all together. So, and I'm really, really glad that Ann and Sriram talked to you about these image-based screens, because that's what we're all about here. And as you can see from the title and the huge red neuron kind of wandering across the screen, we want to try to do this in really some of the hardest cell types that you can do. And I think that's what it's leading to, right? So there's some assays where you can ask about, oh, the cells survived or they died. Great. You could use reporters. But then you have to get into U2S cells, which we absolutely love, and look at their phenotypes. But now what about when you get into cardiomyocytes, when you get into neurons, when you get into organoids, real physiologically relevant things that are just, how do you ask a question about what the variants are doing? And so that's what we're trying to do for neurons, and it means we have to change some of the rules to get that to work. The motivation here is for a whole suite of neurodegenerative disease. Many of these are heritable, so neuropathies. I have a quick list of how heritable they are. A really good starting example for this is Charcot-Marie tooth disease. A neuropathy, CMT2A, is one of the few that's just caused by a single gene. Many of these, by the way, are super idiopathic, right? So different people, they're caused by different genes. This is one of the few cases where there's one gene, MFN2, that's heavily involved. And of course, just like everything else, we want to scan through it and find out all of its secrets. A lot of the neurodegenerative diseases end out with a state like this. Your neurons start happy and intact. And of course, the axons are way longer than I'm showing here. But they end out over time. They don't die, but their axons degenerate, so they can no longer talk to each other. And so to get that phenotype, to understand what's going on at the end of the axon, we really need to look not just at the soma, which is already really hard, but we need to look distally out at the end of the axon. So how are we going to do that in the setting of a pulled optical screen like what Sriram talked about? And this isn't just something that I'm making up. So for most all neurodegenerative diseases, when you look even at patients, when you look proximally, everything looks normal and happy. But as you look more and more distally, that's where the pathogenesis starts to occur. That makes sense, right? The center of the neuron, where the soma is, that's pretty well protected. But as you get further and further away, there's so many concerns and so many, it's insane that they can work so far away from their nucleus. Okay, so that's where we're coming from. So how are we going to do that? Here's some of my partners in crime in St. Louis. And so we came up with this system to try to answer these kind of questions. And it's a little different because, again, the neurons are a little different. Here's what makes neurons really tricky to work with in culture. The axons go all over the place, right? They cross each other, they fasciculate together, they branch out again. If we want to ask questions about what's going on at the end of an axon, but we need to capture the nucleus where we have the guide RNA, how the heck are we going to do that? So, and there's a few different ways that people have come up with. So the first of which on the top there is just to ignore the distal part of the axon. So that is still a fantastic approach. So Daphne Kohler, who started in Citro, this is images from their paper. They're doing an amazing job of this. So they are doing huge scale screens with real neurons, but they kind of have to just kind of throw out the distal parts of the neuron. So that's one way to do it. Another is we'd really like to, and we have a lot of other projects going on with in situ sequencing, like you guys just heard about. But again, how do you do it? How do you ask questions about these more distal components? We actually have algorithms in-house that are hopefully going to get published, pre-printed at least over the summer, where we can start to untangle the axons. And this is not based on Brainbow or TETBO. This is based on some pretty cool machine learning that we won't have time to talk about. But the third is where we've spent most of our time, which is to kind of corral the axons to make sure they stay relatively close to the cell body and not really give them a chance to overlap. And so this is something called a microraft. And so it's not like a little tiny well. it's just a surface, like this grid of patterns that I'm standing on. The cells are just sitting on that surface, and based on the very, very subtle geometry, they tend to kind of stay corralled together, which makes it so much easier to do these analyses. And so our system is called RAF-seq. So very much like some of the stuff you've heard about, very much like cell painting. But now all the cells that are sitting there with all their different mutations are sitting on these different physical grids. And those grids are releasable. And so we can image through it, do our analysis, and say, oh, yeah, that's a cell that's really cool. That's a cell that's really cool. Physically pick that out. This is an automated process. We don't have to do this by hand. We have 3 instruments. So the company is called Cell Microsystems. We have an SBIR with them. We developed this technology to be able to do this kind of functional screening. Very, very slow compared to, say, flow cytometry. Still very fast compared to in situ sequencing. And then, of course, we can pull those cells out. What's neat about this technology is we can really do whatever we want to those cells after we pull it out. So, right, we can pull it out and put it straight in an extraction buffer and do single molecule DNA sequencing to find out about the guide RNA, to look at the cut site. We can do RNA-seq, very similar FACT-seq. But we can also keep the cells alive because the cells are just sitting on their bed, and we're not ripping them off. We're taking out the bed and gently setting it down in another plate. So very powerful to do those kind of things. OK, so that's RAF-seq, and you can see why we needed this to do neurons. Now, of course, day one, we didn't throw neurons on the rafts. We said, let's do U2S cells, because this is an amazing cell type to ask questions about. And so this gene, MFN2, that's known to cause these neuropathies, it also disrupts the mitochondria. It doesn't kill the cell. There's no survival change. And you can't see it by flow, but you can see morphologically that there's fission and fusion deficits. And so we looked at MFN2 and U2S cells. We saw these deficits. And then we used a cell profiler kind of approach to pull out the differences in the mitochondria, these morphological differences. And the first thing we did was to kind of scan through all possible many of the mutations in MFN2, pull those out. That's published. And then recently, we have a preprint that just came up where we've done this across a large library of mitochondria-related genes, and we can ask which of those mitochondrial genes have similar phenotypes, very much like what Sfaram was telling you with, like, here's a normal cell and here's an MFN2 cell. These are all of the other genes that have those phenotypes. But then, of course, it's a huge image-based data set, so we could also ask questions about, in this case, other kinds of mitochondrial phenotypes and do something called anomaly detection to look in different spaces and pull out hits that were different than MFN2, but still very different than wild type. So one last thing I'll tell you about before we dive deeper into the neural space. And again, so you can actually see the outline of the raft in those pictures. So if we overexpose, you can just barely see where the raft starts and ends. These rafts happen to be about 100 microns across. And so you can see some different mitochondrial phenotypes here. And so again, picture this. In a primary screen, whether this is DMS, whether this is a silencing screen across many guide RNAs, wouldn't it be great if we could say, that cell has a phenotype we're interested in. I would like an isogenic clone of it, an isogenic line of it. So that's one of the really big advantages of RAF-seq is we could do this. So in this paper that's up on preprint, we looked at 2 genes that are known to impact mitochondria. Actually, Anne Luria had ATD3A, and I was like, oh my gosh, I've never seen that gene in a presentation before. It's known to cause really rare defects that give people neuropathies due to mitochondrial perturbation. And so we kind of scanned through that. We found mutations through RAF-seq. But then we could also take those individual cells from the primary screen alive, grow them up as an isogenic clone, and now, of course, genotype them and say, like, oh yeah, here's exactly what the alleles are. Here's what the cut look like. But now we have something that we can go and say, hey, I wonder if any of the drugs that are known to affect mitochondrial morphology, I wonder what those do. So right, we have this now reagent that we can share, and many of our lines that we've generated this way are available for you guys to use. But so now for future therapeutic research, and like in Anne's talk, moving from these screens into therapies is a really big thing that we all need to do. And this is one way to make it a lot easier and faster. So this is what this looks like. So when you pop out one of these rafts, Again, the cells are just sitting on the rafts. They're very happy, unlike in flow where they're ripped up and put on a lot of forces. Now, it kind of reminds you like of a pancake with the butter on top. You can see the raft in the center, and then the cells just start to grow out from the raft over time. Another really cool trick is we can use bigger rafts, cells that are really, really hard to clone, like iPS cells or progenitor cells from iPS cells. You can grow a small colony on the raft before you pick. So, right, because then there's a lot of cells around that help them stay alive. And then when you're picking, you're already at like the 8-cell stage or the 16-cell stage. Your chances of growing that into a full isogenic line are much higher. OK, on to pulled optical screen with neurons. So these are real neurons. These are human, or not primary, iPS-derived human motor neurons. This is a month-long protocol. It's not the NGN2 DOX-inducible protocol. There's many good uses for that. So there's a lot of development there to get that working, and then also to get it to work in these high-throughput scalable formats. It's just so beautiful to look at these individual neurons. In this case, you can see the little puncta of their mitochondria. You can see their axons. But across, it's like a 6-well plate that's subdivided into all these rafts. You can imagine there's just tons of heterogeneity. So Purva and the lab solved that problem by coming up with a classifier. You can see on the bottom, it ended up becoming quite complicated, right? Just to pull out, here's all the examples of the cells that we care about, right? Here's good quality neurons. And there was cases that were just tricky, right? Where there's a soma over here, but a neurite coming into the same field. So these classifiers are able to sort those apart. All the data for this is now on Hugging Face. So if people want to look at tons and tons of pictures of neurons, that's there. So we've got kind of all the pieces together. And generally, the kind of screens that we've been doing since this has started to work about 3 years ago, there's kind of two varieties. So the one is the, let's take something normal and break it variety. And then the other is the inverse, which is what I'm showing here. We take a cell that has an ALS gene, for example, in it, that it's already mutant. It has slightly perturbed distal mitochondria. Now we put a pulled library on it. And then we say, can any of those drugs actually rescue it? Right? So we can go both ways. We can try to break things or try to rescue them. And this picture I've showed down here with all these addresses, I didn't say it out loud, but again, the way that the raft system works is that since every cell is sitting on one of these little tiny releasable rafts, they'll have an address. So we can just make a list of, here's the addresses that we want to physically pick, feed it to one of our 3 machines that do this day after day, and those cells get physically picked out. And we can, again, do whatever we want. Usually we're asking what guide RNA or what cut, what genomic consequence. And so that's the kind of data we've been working with. But these are neurons, right? So let's talk for a second more about these phenotypes. I've been saying mitochondrial morphology. You guys now know about cell profile or about making classifiers to distinguish different kinds of nuclei. How are we doing it here? And this is kind of the set of tools that we usually use. So you have a complex, usually single neuron. I'm showing a few here just for fun. And then there's a few different approaches here to get to features. So on the very top is like cell profiler, right? So this is something that uses the pixels to generate, like Anne said, she could describe every single of the 1,500 features to you because it's like, well, we're going to count pixels that have a certain intensity. We're going to look at the textures, right? So those are good because they're very interpretable, but they're still based on pixels. And one of the big problems is if there's not a feature and you need to measure that, then you're out of luck, right? So for example, how do I measure in a degenerated axon where the pixels don't even connect what's going on with the mitochondria in a distal area, right? There's no software that has a feature to do that. So we can't use that. The other one that more and more people are using is on the bottom here. I have kind of an old-fashioned network drawn for you, but the idea of autoencoder, it seems silly on the surface, right? I'm going to give an image of a cell, and this brain in a computer is going to give me back the same image of a cell. Why would I care? Why would that be useful? Well, the reason it's useful is because you can teach it what a cell looks like, and it actually gains the ability to draw a cell on its own. And the beauty is that you're forcing it to learn by squishing it down into this latent encoding layer. Again, they're actually much more complicated than that, but that's the idea. And so they have to figure out with very little information how to reconstruct that. In the process, they learn about how cells work. And so you can use the middle, the lane encodings now, as features. And I'll show you how it looks in a second. But the most interesting is the second line item here, which is called a physics-based reconstruction. So proof of principle here. This is a member of the Bork complex, block we'll call it, and it's involved in actually bringing, JT said I have till 5 p.m., so we're good with time. No, I'm just kidding. This is involved in transporting both mitochondria and lysosomes across the axon, right? So one thing that's just very easy to use as a test case. And so when you look at these in details, sure, you can see mitochondria along the axons, but every neuron is different, right? And the morphology is very complex. So here is that, these are some cumulative histograms. And so basically on the bottom here, you're seeing one of these autoencoder features. This is from a publicly available autoencoder that you guys can all use called DynoVIT. And it just happens to be that there's a feature within the autoencoder. So something it's learned about looking at pictures of things separates out and can tell the difference between these cells that have basically more mitochondria distally and fewer proximally. And it can find that, but it doesn't do a very good job separating out, right? You want more and more separations. So basically, this is the coolest part. We now have these physics-based approaches where we actually, that's really not a neuron. That's completely simulated. And in the simulations, we can, again, teach these deep learning algorithms what it means to be a mitochondria at an infinite scale. And so it actually can understand, like, here's mitochondria. So this is not a pixel level measurement. This is at the scale of, like, angstroms. And so when we do that and apply it to this data, what you actually get, oops, sorry, pressed the wrong side, is really, really good separation, and it's meaningful. It's not meaningful at the level of pixels, but it's meaningful at the level of biology, because you actually say, oh yeah, there's a mito-size ratio difference along the neurite, and it goes in the direction you expect. And so these are the newest set of really powerful ways to get biological insight from the images themselves. And so we've done that with the screen. It looks awesome. I'll zoom out because Doug's getting closer and closer to me very creepily. So that's the overview, and we've been doing this in spatial transcriptomics as well. So I hope you guys have lots of questions for me. Thank you so much.
We'll have to take questions after.